this, I think you've had this, but the other day, you know, that's part of being live. I'm kind of gauging your responses on things, and if I feel like you need a little refresher, I want to do it. We've talked about genetic engineering to some degree and recombinant DNA. We're really talking about, you know, when we are putting a piece of DNA into a plasmid. That's pretty incredible stuff. I mean, we take it, your generation, and now our generation, I guess our generation on Earth, you know, assumes that we take it for granted. You know, we make copies of DNA, billions of copies in a few hours, and we can sequence stuff like, you know, as fast as jet engines can turn. You know, it is amazing. And I just want to briefly go over 3 or 4 of the principles that people discovered that essentially put this into our heads. And as I said, probably you had this in general, I'm guessing, knowing Dr. Bonica, but maybe in other places. But let's just go through a few things. There's essentially 4 topics here. It's a very short lecture for you. The transformation. Okay, I'll have a diagram here, but for those people who respond better to words, I'm a picture person. I sometimes have hard time saying things and hard times writing things because I see things in pictures. But so we have both. A process in which donor DNA molecule is taken up from the external environment and incorporated into the genome of the recipient. Okay, this was one of the first ideas, once they figured it out, that, wow, there is recombinant DNA. That's, you know, DNA can combine. What does that word mean? It means you can recombine it. It can go in and become part of the genome. You know, so first people started to figure out DNA. Okay, the early parts of transformation were figured out before they figured out the structure of DNA, but they didn't understand the mechanisms. They understood the effect, which is cool experimental biology. And then when people started to think, oh, then there's this genome, and they started looking at us. Oh, there's chromosomes, and they started putting this stuff together. But then folks essentially thought, well, our genome is pretty fixed, you know. And then they started saying, well, you know, it looks like not everybody's the same. It can't be too fixed. As well, meiosis is where things really get mixed up. And you know, there's the daddy and the mommy and those chromosomes mixed up. And then they started finding out, well, you know, some of those chromosomes kind of cross over. And so, you know, for science, who loves to have, you know, many of us are here because we love it because it defines it. This is the way it is. Look at those molecules in the elements in the periodic table, nice square in rows and columns, and it all means something. We love that. The genome, you know, it kind of moves around. Is that right? Can that be true? Yeah, it is. You know, it is. So probably a refresher, but probably everyone comes back to thinking about Griffith and Fred Griffith and his transformation experiment, and he's doing this in the 1920s, 1930s. Well, it seems like a long time ago. He was a nice guy, dressed sharply, like dogs, so he's a nice guy. But he was working with strains of bacteria. Now, oftentimes people start mixing up bacteria and viruses, and it's easy to do. Just I'm going to try to help you through that. Okay, and he's got 2 different strains of bacteria here, pneumonia. He's got one that when you look at it, and you look, and some of you have seen this on the petri dish, you notice like bacillus is kind of waxy and oh, I don't know, a little more stringy, and just even in the colony, and you'll see these other ones that are kind of really mucoid and so forth. So he's got cells that both have, that look mucoid as a colony, and they look smooth under the microscope. Microscopy is doing okay now in the 30s. It's not quite as nice as what you have, but still pretty good. And so he's working with a disease. He injects these into some poor little unsuspecting mouse, and this mouse expires. Okay. Now he has another strain that's alive, and it's the same strain, so same species of bacteria, but it doesn't have this smooth capsule around it. Okay, there's something in that capsule that causes pathogenicity. And he puts this into the mouse, and the mouse is like, ouch! But then it's like, okay, and he's all right, stays alive, and that's good. This little insect is saying the same thing. Pathogenic strain with the smooth, most is dead. Pathogenic strain with the rough, and harmless S is the rough. They're kind of square there, and most is okay. Then he takes the smooth ones and he heat kills them. He basically denatures the bacteria, so the bacteria cannot express. Okay. And he puts that into the mouse, and the mouse lives, and here they're checking. He said, oh, yep, really did kill all those things, couldn't get them to grow where I could get them to grow before. It's a little check that he's doing, you know, kind of a holdover from Koch's postulates. Okay, and then he mixes the harmless roughs with the heat-killed smooths. You know, so theoretically, what should happen there if you inject that mixture into the mouse? Basically, is mixing two of the strains that should allow the mouse to live.
They should stay alive, right?
Hmm?
They should stay alive?
The mouse should stay alive. I mean, if you're going through just regular deductive reasoning, you know, and saying, well, nothing's happening. If I do this, mouse dies, so red one. Okay, blue one, or purplish, violet, whatever, mouse lives, okay? If I take this one and I heat kill it, mouse lives. So let me take the heat killed one and these guys. Let's not worry about this one. So I should get 2 live mouses, or mices. Okay. And then he puts in mouse dies. Whoops. Okay, checks it out, sees not only just that the live cells are growing, but both cell types are growing. Okay. Griffith didn't understand, but he had found DNA transformation. Something from the heat-killed pathogen that should not really have done anything, something came over here, incorporated, and allowed the live cells to express, and vice versa. So he saw the effect. He got it. He said, well, you know, something moved around. And then, you know, in that time, he might say a lot of, you might say, well, it's the surface. Maybe they didn't denature it, or it's this, it's that. And it took a while to figure out then that it really was DNA moving between the cells. And once again, he was pretty lucky. To have transformation work out, it's best to have competent cells. Okay, competence means to become competent in picking up DNA. All right, so we can think of it as having kind of leaky cell membranes, and of course this was a gram-negative, a leaky outer membrane in the cell wall, and a leaky cell membrane, outer membrane, and leaky cell membrane, and it's able to pick things up. That's cell competence, a little bit different than competence for the MCAT or some other thing. So that's what's going on. We had talked about transformation. I wanted you to have a feel for that. You can do this, and maybe some of you did. Have any of you made the, one experience really popular, I used to do it in art class, I love it, is you can take regular coli cells and you recombine, you pick up a plasmid, and you make the cell glow. Anyone ever do that experiment? Yeah, they're going, that's kind of fun. And everybody like, oh, it glows, it gets my attention. I love this thing. So, yeah, I like stuff that glows too. Oh, yeah, and that's one of the isolates we can do for micropunning. There's luminescent bacteria that live in fish slime, and we could isolate those. So they're glowing bacteria. We can do that again. We won't transform them. Anyway, so, yeah, you got to have his confidence, and you can do it several different ways. You can use electricity. And you put a certain concentration in the DNA, whether it's a plasmid or a strand. Usually they use plasmids. And you can kind of electrocute them, persuade them. Okay, I'll pick up the DNA. Just stop that. Or the most common way, and probably the way you did it in General Micro, or wherever you did this, is you put the the cells in with the plasmids, you let them kind of hang around for a while, and then the plasmids start to stick to the outside of the coli. And what you do is you heat shock. You go from about 30 to about 42 degrees is what I recall. And that causes the cells to expand, so they'll go, kind of like that, physically. They won't breathe in, of course, but like that. And they'll bring in some DNA, and miraculously it recombines. So isn't that pretty neat? I think so. So what do we have here? We have the exchange DNA. We know we got to talk about complement cells, at least be aware of that concept. We know that DNA strands can work, and plasmids could also work. And plasmids will tend to be expressed and put into the proper cell. What's unknown? Some of you guys like thinking about the bigger picture. I read a paper a few years ago where people were saying, transformation is a cool concept. I mean, it occurs outside of the flask, and these groups of researchers, this group of researchers was saying, I think that transformation might go on more than we think. It's possible that lightning strikes, and they had how many lightning strikes per square kilometer or whatever, and they were thinking that lightning might cause recombination naturally. Never saw a follow-up paper. I have no idea if that ever went anywhere, but people think about stuff like this. I thought that, sometimes having, seems kind of like the preposterous. It's worth considering for that. Now, number 2, transduction. Here the key is to look for a virus. Okay, and the virus that we're going to be concerned about is one that's able to do the lysogenic pathway. So we're talking about a temperate virus. Now, all those terms, all those nouns should mean something on some level to you at this point. So, in transfer of gene, best described we're going to stay with bacteria, so bacteriophage and bacteria. The virus acts as a vector to move the DNA around. So, T1, T4, those phages that are double-stranded DNA are great for doing this in the best study. Okay. If the host bacteria survives, if it doesn't go into being the lytic cycle immediately, it is transferring that DNA to subsequent generations. Okay. Now, this is all written down for you. It's on blackboard. So I'm going to move ahead and just for the interest of moving things along. There are 2 ways transduction can occur. Okay, let's take our focus to generalized transduction. You can read those through those as good as I can. Basically, you have an infection. The DNA is mixing up. Things are being expressed. We've kind of been there, done that in this class. You know about those things. But then, when the bacteria is, in this case, it does finally go through lysis, you have a mistake. Rather than it only picking up its viral genome, it picks up some of the hosts. Okay. Is it a mistake? It's hard to say if viruses, who knows what's a mistake. But, we know it's down to it's a probability game, right? So, and there's so many of them, and there's so many bacteria, just like when we get genetic antibiotic resistance. Not all of them are resistant, but it only takes a few. It's a probability game, right? And conditions. So anyway, this guy can bring this allele over here if it aligns up with the allele, the opposite allele in the genome of the host. It's expressed, and so you've moved DNA around in between generations. In specialized transduction, it's basically the same idea, except that it's picking up a nick of the DNA, so and bringing along for the viral host, and then incorporating it. So it's a little bit different, but the mechanism that we need to know is about the same. Viruses are moving around DNA that is not their own, and that's getting incorporated into the genome and possibly moving genes or gene expression around. Okay, especially if you have allelic expressions. Okay, the second and last one here, conjugation. Sometimes talked about in the more relaxed literature as bacterial sex. The only thing close to sex on this is that bacteria do have a way of contacting each other, but there is no meiosis. There is none of that stuff that has to do with sexual reproduction, as you've all learned it. So the world is safe. You're you're okay there. So I'd rather not use that. But sometimes you read that and it sometimes confuses as much as it helps you remember things. So, okay, the temporary direct contact between two bacterial cells leading to exchange of genetic material. And this will be, we're talking about DNA. This exchange is unidirectional, they're not going back and forth, okay, so in a way, it seems kind of sexual. You have a donor cell and a recipient cell, but that's about where the parallel ends. So, and this is the implications. You can possibly move things, information around, and it can be expressed within the generation, so it's horizontal, as opposed to waiting for the next generation and recombination of things to be expressed. Okay, so conjugation typically is thought of as a plasmid. Okay, so we have this plasmid in the donor cell. This pilus is the conjugal pilus. Pilus can also just be used for attachment, but some are specialized. A little protein sheet kind of a thing, a tube. Okay, and we have the receiving guys over here, and they make contact. And kind of what we saw, that this rolling DNA replication that we talked a little bit about with the viruses, the M2S, I think it was. We see this with the plasmid. Plasmids are good at taking part of the template, peeling it off. They can replicate that DNA very easily back in this machinery, and the DNA moves. Here they're showing that this is occurring bacteria to bacteria. There's also documentation that it can move through the conjunctive pilus. Some of the things to realize, antibiotic resistance can be moved around this way. People have shown that, although other mechanisms could work. Transformation itself could work. Okay, you could get, you could cut that tube somewhere in the process of transferring, so you might not get all the material exchanging and so forth. As far as its prevalence in nature, most of what I've seen has been in cultures. I don't know how much this actually works in nature, but because it's described in the laboratory, I'm sure it does. It's just we know so little in some ways about bacteria in nature, it's hard to know how much of this goes on. So it's easy to see then transformation, leaky cells, competent cells, moving DNA around. The plasmid could be moved around that way. Transduction in viruses, conjugal pilus is what I would emphasize in our class over this, because this can be confused with, in a way, transformation. And then that's pretty much that. This, I'm not going to go through this with you. This is a little summary, so you can look at them side by side when you're studying. I thought, oh, that might be fun for people to have. And a few notes on that, trying to distinguish the three. So sometimes people get them mixed up. So just find some kind of word play that works for you to keep it straight. And I think that's about that. I did want to put in this person. I love role models in science. Okay, for me, male, female. If you're doing great work, that doesn't. It's never a thing for me. It's like, oh, I only listen to men or something. And if you got a great idea, so that's why I was talking about Margulis, and I talking about Jenner, and Griffith, all these guys. They're amazing. Barbara McClintock, okay, just relatively recently passed away. Started as a young woman over at Cornell. Got into corn genetics from the get-go. But look at some of the stuff that she's figured out. Okay, she started to look at corn cytogenetics. Okay, that's cell genetics. Okay, describe genetic recombination crossing over. Oh, that's the person that figured that out. Okay, she's really active in the 30s, 40s, and 50s. Okay, so demonstrates the role of the telomere and the centromere. What is that? You ever hear of a telomere and a centromere before? Not in bacteria you haven't, but where'd you hear that word before? It's in there. I know. I can see you going, oh man, I haven't thought about that word. So say it a little louder. I'm here like, who is it?
Eukaryotic chromosomes?
Yeah, so that's exactly right. And where would I find those on a chromosome?
Is it meiosis is when they appear?
Yeah, well, they're, yeah, when they condense, after you've made the DNA and the chromatin wrapped around the histones and going into the nucleosomes, after you have super coiling of that whole complex, you get the classic X thing. So the telomeres are the arms and the centromeres, the connector, and well, they're actually, would be more like this, I guess, because they separate. Oh, my arms is chromosomes. Anyway, so okay, so she's figuring that stuff out, and then developing theories explaining suppression and expression of genetic information from one generation to the other, how genes can not only turn on and off, but then how genetic information can move. And you may have heard the term jumping genes, very popular amongst people talking about that, transposons. So here's two different mechanisms: cut and paste, pick it up, paste it in between a gene. Oh, that could really affect the expression, right? You know, especially if you got the promoter and stuff still in there, and now you put more, something different in the middle. Okay, that's kind of neat. And she's always holding the ears of, we used to call them Indian corn. I don't know if it's politically correct anymore, but whatever you call it now, corn that has different blues and reds and stuff, and yellow kernels. And she's just, most of them go, oh, these are pretty, I'll hang them on my door. But she's saying, well, if you look at the way that these numbers work out, it doesn't work out, like by like the chi-square expectation. There's other things going on here. And so she's saying, well, stuff's going on here. I think it's transposons, although I don't have a word for that yet. I'm going to discover that. And so she does that. And then they talk about then the copy and paste mechanism. You can see very easily it's a little bit different. But I'm not going to ask you to draw that. No transposons, no McClintock, I think. I think she's worth knowing. I certainly think she's worth knowing. National Academy of Sciences. I'll never make it there. Perhaps some of you will, but Nobel Prize. Oh my gosh, we should pay attention to what she says. So that's it on this little bit of a commercial on genetic movements before we move into evolution and other stuff. Are we good? Okay, if you're good, don't say anything. Good, all right. Now, if you're not good, yell, throw things, I'll listen. Okay, we kind of did that. Evolution, great. What happens when you stay up for a few days? You start bouncing. Hopefully I won't be too crazy. Okay, systematics and evolution. Systematics is how we classify things. Evolution is what? What comes to mind when, this is like the central theme in all your classes, certainly through the core. What is this evolution thing? Tell me something you know about it. Yeah, right. Those kinds of questions, don't they just kill you? Evolution, that's a big thing. Well, tell me something you know. Well, what comes to mind? Well, we have people who are very unhappy with the idea of evolution, okay? So we're going to talk about, on a science level here, theology is a different place on campus. We'll talk, argue about that, different. We'll all be respectful of each other's opinions, but this is a science class. So the science of evolution, give me something that you know about. What are these scientists talking about? Anything.
There's like selective pressures that like help enforce or unenforce a different trait.
Yes, selection, selective pressure, okay? And we talk about things like survival of the fittest kind of thing. We talk about reproductive success. We might think of success more in monetary or something, but if you're not passing on genes, you're not really, as far as your population goes, you're not playing the game. I mean, I'm an evolutionary failure. I will not be passing on my genes in generations, and that's a whole different sort of thing. But hey, failure, get used to it, right? Like they said in The Princess Bride. So the thing is you have selection for things. So it's a probability game on some level, but a probability game that works out with genetics and genetic recombination, and also works out with the environment. Okay, name me a person or two that was a big player in evolution. This should be easy. Darwin, Chuck Darwin, right? Charles Darwin. born at the same day as who? Abraham Lincoln. Yeah, we said that to our freshman class. Jim and I used to teach general biology together, and he put that out. He said, Charles Darwin was born in 18, the same day as who? And they said, as you, Jim, which I thought was hilarious. I still laugh at that one. But, and we laughed at the time, and he's got a great sense of humor. So, yeah, as Abraham Lincoln. So that puts you, gives you a little bit of perspective on that. Okay, so we got some idea of evolution. We can think of a few other things as we go along, but what we want to think about with microbes is we have some hard questions. We don't know exactly when microbes die. We don't know how many species out there, and we have trouble even defining what a species is when it comes to the prokaryotes. Now, all of you, as biologists, in a third-year course, a 300-level course, have learned a definition for species. So help me with that. What have you learned? What do you remember? Even if it's like, if it's nothing, well, let's move on to what we do remember. What can we define a species amongst us? Go ahead.
Like the most specific you can get when classifying.
Yeah, it's the definable unit of biology. Okay, when we get into families and orders and classes, people are making groupings. Okay, anyway, even if you do it genetically, and so different things are being weighed for. But when it comes down to what is it, species is the least common denominator. So well put, very very nicely put. Okay, I like that. But now we know what it is as far as helping science. What is a species? How would I know one? Go ahead. You first, and probably you next.
Things that can reproduce, actually, things that two organisms that can reproduce together and produce viable offspring.
There you go. You can have sexual reproduction and there's a viable offspring. Thus, the inheritance of the information. Good. Were you going to add anything, Jake? That was pretty much it. And, go to the back of your book. That's pretty much what it's going to say, except it will get into the prokaryotes because Madigan is a stickler for details. So, what's the problem with what we just heard here from Ashley on how species works for the prokaryotes? Let's just say bacteria right now. I was thinking of archaea. Who knows about archaea? Yeah,
Because bacteria don't have sex?
Yeah, they don't have sex, So, binary fission, sometimes sporulation, they're budding. We've kept it simple so far. But, yeah. So what you do, well, that species thing goes out the window, and you gotta say, well, wait a minute, hang on, what do we got here? We got the most fundamental unit in biology that tells me that that's a pretty important thing, okay? And then I got Lemke over here saying, 90% of the species on Earth are prokaryote. Wait a minute. If you can't define it, how can you say that? You could say anything. It could be like a presidential debate. What are the facts here? Well, it is a quandary. It is. Talk to Jim and anyone you want about it. People do have trouble with this. And we've come up with the solution already in this class. We've talked about what molecule is going to be our clock, our comparison molecule.
Genes.
Hmm? Yeah, genes in general. Yeah. So in like I say, your generation, your kids will be doing whole genome comparisons. It's already happening. Okay, but right now we're still going to take one gene and we're going to use that across everything and see if that clock gives us a tree that looks something like the tree that we make with comparing traits. What's that gene, or where does that gene come from? Give me something, Jay.
16S.
16S in the prokaryotes, 18 is the small subunit, small ribosomal subunit rRNA. Remember, there's proteins around all that stuff too. But we're concerned about that gene, which doesn't really transcribe or translate into anything. It doesn't make a protein. But it's very important because part of that's, so think about it. what characteristics of that gene makes it such a great clock? Well, everything has it. We'll have a slide on this in a minute, but the other thing to think about is that its function, because it has double and single stranded areas, is to really make that small subunit, get with that large subunit to do what? What's the ribosome the site of? Translation, protein synthesis. And we're going to say, well done. Okay, so we know that. So we know to some extent, conformation and the shape of stuff is really important in biology. People taking biochemistry are probably painfully aware of that because you talk about enzymes and kinetics and stuff. Okay, so parts of the ribosome then need to be conserved to keep that shape correct. And parts of it can be relatively variable. The trick is to see if you've got variability in the sequence, is what I'm talking about. Okay, so in that 16S sequence. So you need a clock that over the time period that you're measuring, you have enough variation that it keeps things recorded in that clock. Okay, and the way we see how the clock really works, I'm talking about a clock that records change, is that we develop these phylogenetic trees that show common ancestors, and we go, oh yes, it looks like the archaea branched from this thing here. These guys branched from here, and oh look, the proteobacteria, are here, and the mitochondria is in there. Oh, that matches up with a theory that we've heard about, and so we gain confidence in this theory. Before that, the kingdom model was great. Looking at traits was really good, and what traits were we able to look at the most? Plants and animals. People used to think biology means plants and animals instead of life, bios. But it's this plant. And we looked at stuff that was big and bigger, and we've kind of gone through some of the history on that. Well, let's get into this. Now we have a bit of a preamble. So some things we do know, to put this in perspective, we're talking about, on Earth, more zeros is even in my paycheck in dollars, okay? 1 to 5 times 10 to the 30th. Now, at first, it's only 30 zeros. Okay, but you start doing that powers of 10 thing, sometime. Just do it sometime. Get a sack of rice or something and do 1, weigh it and do 10 and start, all of a sudden you're up to 1,000, 10,000, you got big bushels of it. It's pretty easy to say that when you scale up, this is a lot of things. So we know microbes are around. We've talked about that in opening days, Species, gotta tell you, it's out there, okay? 4,800, okay. E.O. Wilson. Anyone know anything about E.O. Wilson? Here's another person I'd like you to know something about as a biologist. Anyone? E.O. Wilson. It's okay. Don't feel badly. Now you're going to know something about E.O. Wilson. E.O. Wilson grew up in Alabama, and when he was young, he had an accident, and he actually lost the use of one of his eyes. What was really interesting about that is he went into a life of looking through microscopes and so forth, and he goes and figures out the whole field of sociobiology. Yep, he's the guy that figures out what the ants are doing and what the bees are saying. This whole thing of the interaction, living in swarms and in holes and all this. He's figuring that out. That's E.O. Wilson. He's alive. I got to meet him one time. Thrilled. So he also came up with this concept of really promoting biodiversity. You guys know about that? Give me an idea of biodiversity. Somebody. Jay, take a rest. He knows all this stuff. Somebody else. And good for you, but biodiversity. Put the words together. What do you think it is? Or the parts together. Wow, you should, okay. Amy's going to be real angry at you guys in ecology if you don't know biodiversity. Okay, go ahead.
There's all different species living together.
Yeah, this whole idea of these species and the importance of all these species on Earth. Okay, a simple food web compared to a complex. Stability, resilience, those kinds of things that come up when we study ecology. So E.O. Wilson says, when he writes his book in '92, great book, he writes 4,800. he's basing this off of defined species that were culturable. And E.O. Wilson was not wrong, but you know already then that this must be short by a factor of a few hundred, a few thousand, right? Because we, you know, if we go into the culturable is only 1% or less of what's out there, we know that he's grossly underestimating, but you know, he's working with evidence. He's a Harvard professor. He keeps things as real as possible. Before I move on, just want to say a great book, if you want to read a book about, you know, someone who's been there and done it, and still a nice guy. You know, he says even as I grow older, I appreciate manners and, you know, and interactions even more so, because that's the best way science is done. I can just tell you, science can be very cutthroat, believe it or not. So anyway, The Naturalist, very nice book. And he's also just written another one, Letters to a Young Scientist. If you want to get advice from E.O. Wilson, he's written a nice book on it. Very short, you can read it on the bus ride in or whatever you do. Okay, Torvik, okay, 96 also, the same time period, okay. I was doing my first postdoc. I'm reading Torvik's work, and he's saying 10,000 species per gram of soil. How is he saying that? That guy's crazy. He was doing DNA to DNA hybridization. He was extracting DNA and seeing how much from different samples and seeing how much it matches up. If it was perfectly matchable, all the DNA would hybridize, the other DNA would be perfect, 100%. He started saying, well, I get about this much, and he did another something, I get this much. And he started doing the math of probability, says 10,000 grams. People do not argue that. At the time, they did a little bit, but now they're going like, Torvik might have had something there. Okay, importance of bacteria, we still don't know. We know that bacteria now shape our viewpoint of what we've for so many years have taught and learned, that the base of the food web, any food web, this could be rabbits and foxes and grass, but that the primary producers have the biggest biomass, and because of efficiency of consumption between the different trophic layers, we get less and less of the higher order consumers. Then we put bacteria down here. Okay, are these guys really eating them? No, they're eating the nutrients. What are the implications? Not quite known yet. I've only seen a few papers really trying to tackle with that question. It really came out in about the last 5 or 6 years. So, here's probably the level most people are at if we're lucky. And probably you coming into the class. We're probably around here. We can look at our microscope at about 1,000x, and we see some spots. Okay, and these are nice red spots, and these. It's a little bit of a pixelated picture, but we see purple spots. Some of you have been showing me your purple spots that if they're clustered, they're staphylococcus. And this is kind of our understanding of this on a lot of levels. So diving into, in this class, into what it means to be a species and then all the biodiversity, it is an amazing trip. It'd be like no other class you take. You can sit and learn all the plants, you can learn all the mammals, all that stuff, and it will be a lot. I've taken many of those classes in my undergrad also. That's what I did. My undergrad's in wildlife management. I had to know what I was looking at. But bacteria will blow your mind, and will hopefully, in good ways, and we'll talk about that. The other way that we understand bacteria, and we have done so through the ages, and probably first, is we thought about disease. Okay, you forget to brush your teeth, this is what happens. Okay, you can get dental caries, you can get cold sores, you can have bioterrorism with or without anthrax, but anthrax can cause a topical sore, it can also mess up your lungs. People burn whole herds of livestock if anthrax is there. It is the danger. And we have things historically that come out of anaerobic bacteria. Really? They're anaerobic? They don't, what do I have to do? We have poisons with tetanus and botulism that affect the nervous response to such a degree that the muscles can go into tetany. You cannot relax them. They only fire, fire, fire. And you can have a horrible, terrible death as this poor person did. Usually, succumbing finally to suffocation because the diaphragm won't relax and therefore you can't breathe. And the really crappy thing about this is what's going on with your brain. It's still working. So other things, diphtheria. So people saw disease. We go back to pasture before those guys who actually said, I've had it with this disease. I'm going to try to figure something out. These ideas. There's got to be something behind it. So that's probably one of our oldest ways of appreciating bacteria. And it was kind of these germs, this disease, pathogens. So our society's pretty much feeling about bacteria is kill them, okay, kill them all, okay. Let God figure it out. So kill them all was kind of the idea, and that's what we tried to do. And now we're saying, hey, if you can't kill them all, some get really resistant, and you shouldn't kill them all because maybe there's some good to it. So we've had, oh yes, tuberculosis. What would a slideshow be without kiwi? Okay. So we start to see a transition, and maybe what surprises me more than anything else is how long it took to come out in this. When are we talking about disease? People start hating disease from the moment they started feeling bad. So I mean that goes back to probably to Africa. I feel bad. It's got to be something. Okay, but we start seeing that there's a whole other world out there, and this world can be used for other purposes. Science. What is science? What can you tell me about that journal? I have never been published in it. I can tell you that. I would love to be, but it's not looking too good. Too much time teaching. I like teaching you guys. Science. Tell me something about that journal. You should know about this. This is okay to know in your field.
It's like the most prestigious journal.
Most prestigious journal. Science has a turnover rate. If you submit an article, it'll be accepted or rejected probably within the week. It is amazing, and they're all peer reviewed. Now, when you read science, whether it's online or in print, you will get, it's hard at first, you read it, they have a lot of journalists reporting things, okay, but they'll always carry the primary literature also. So, science journalists guys are good, but they're not the scientists, so make sure you know that. But the other big one, published in England, is Nature, Science and Nature. Didn't hear that. So those are really big ones. Short articles, long articles, great. 1997, an issue, Frontiers in Microbial Biology. Didn't we cross all those? No, some of you guys wrote some great essays on your perspective of the history of microbiology. A few years later, 5 years later or so, environmental microbiology. Oh, makes the cover of Science the whole issue dedicated to, which is a big deal. Usually it's something like AIDS or some new gene marker or something. So, people are recognizing that there's more out there than we realize. And there should be one more. Yeah, there it is. Microbial worlds, okay. Now 2008, probably some since then. But what is going on here? Why did this make the cover? Just because it's pretty? What is it? What does this waterfalls have to do with microbes? You guys looking it up? Or just reading email? Okay. What do you think this has to do? Where have you seen something like this, those people in 346, in your life right now? Your young, wonderful lives have noticed something like this.
It's like the halophyte archaea. It's like the halophyte archaea.
It is. We talked about halophytes and how they have bacteria rhodopsin, and then they can be colored. Now this is a freshwater stream, okay, somewhere, I'm not sure the location. It looks like it might have been a quarry, but I'm not sure. It could just be a fault. What does it have to do with microbes? What's going on there? Where have you seen different colored microbes? When Winografsky comes. So we can probably guessing algae. Okay. This does look like a mineral deposit, but it's a mineral deposit that's moving along with iron oxidation. If you go, and around here you see it too, but certainly if you go out east, around mine tailings and so forth, you'll see big orange pools. And we'll learn that that's from Thiobacillus desulficans, acid mine drainage, okay? I'm not saying this is an acid mine drainage area, but there's bacteria that are very good at oxidizing iron. We were noticing, anyone last, I can tell you the story. Walking back to HSB, they had there's a sidewalk, right? You know, you guys talking about stuff with chalk. You get through that, and there's a board, piece of plywood, and there's water running out from underneath it. Anyone see that? Okay, good. Did you look real closely on the top of that by any chance? Did you kind of notice it was kind of shiny? I mean, besides the water being reflective, there's kind of a surface to it. I don't know if it's still there. What that is an iron oxidizing bacteria called Gallinella. And it needs the organic matter that's coming out of the lawn. But it also needs an area that's kind of anaerobic. So the oxygen is being eaten up, because that's how iron is more dissolved. But it needs oxygen, so it makes these little projections. Now let's it, okay, I'm being a Gallinella now. Let's it float on top of the surface area. So at first, it looks kind of shiny. It is first grabbing iron. You are seeing like a very thin sheet of iron. You come back, and I didn't check it today coming in. I'm thinking about lecture. But if you see these in nature, they'll turn rusty, just like this. So iron oxidizers are around. And just like you're saying, like, kind of interesting. I don't know if I'm that interested in it, but there's more stories like that. So people start seeing, wow, there's a lot of stuff out there and all around us. Okay. So why do we want to study bacterial diversity? Because the diversity story is incredible. Already, I asked you a question on the exam. Probably will ask it on the final or the third or fourth exam, however they add up. Because you can add to this story. Okay. You can add evolution and systematics and all these adaptations now. You know about size and physiology, But that's a story. That's a question I can ask you from day one, and you can do a nice job. But I really expect a different answer on day 15th week, whatever day that is. So you can do that. Your eyes are open. You're listening to things. You're reading. You're understanding the stuff. Okay. We have some themes. Oldest life forms. Okay. Now, old isn't always just a deal. Okay. It just means that different populations have been able to adapt, and the earth has changed, and places that were everywhere, like anaerobic earth and hot, smelly earth, are now a few places. Geysers and those places. So those microbes, most of them made it. Maybe some didn't. They have refugia, places where they can live and do things. The oldest life form, it is arguable that it is possible that the first bacterium is still alive, because people don't know exactly when they die. Usually you look at the cytochrome system if there is one. But who knows? We will never know the answer. Well, I'll never know the answer to that. Maybe you guys will find out. Vertical, okay, this adds to diversity, right? A little harder to explain, but there's a few examples we can look at. Symbiosis. It kills you. We all talk about, more species are dying every day from the rainforest than we'll ever describe, right? You guys hear that one? You heard a form of it. We're losing a lot of species faster than we can ever describe. But I was trying to set up, help, at the American Museum, they have a big tanks of liquid nitrogen where they're collecting DNA from all over the world. It's a cryogenic center. It's called the Monell, if you want to look at it online. I'm not messing with it. So, I was talking to different biologists trying to set up a collection center for communities. Because where do we keep our type of communities? And they said, well, mud and air and intestines, that stuff's all good. But they said, think about these symbiotic relationships. You start losing a certain parasite or a certain pollinator. Also with those, you're losing really unique species you'll never see before. How do you get around that? You don't know. But those are some pretty interesting questions. So even though we can look for stuff in mud and soil and our earlobe and everything else. There's things that we just commonly aren't even going after. So when I think about diversity, symbiotic relationships can be really, really important. Mycorrhizal associations, we only thought were between plants and fungus, which is what a mycorrhizal association is. We thought they were unique. They happen to. Now it seems that pretty much every plant has its own mycorrhiza. Well, how many species of plants are there? Now, and they all, so the number just keeps increasing. Okay, are we done with this? Oh yes, and then our roles. Function. Along structure, function is always a theme in biology. I think you do that. What's he saying? Okay. So, quickly, kingdom model works for us. I don't think there's anyone in here that couldn't name the 5 kingdoms at this point. Yes, there are 7 kingdom models. What did I say? 7, yes, 7, 6. You may have learned, and it's still out there. I think even Margulis likes this idea. She doesn't call them the Monera. There's the eubacteria and the archaebacteria. And people now say, no, they're not bacteria. They're completely different domains, and that's the way I teach it. But you might see that as a model that makes it a 6-member model. These things happen. So, all that, the protista. The protists are so hard to define. If you had to define what a protist is, I used to put this on the first exam and the last exam, and I never got much different in answer. It's hard to explain. How can you define a protist? It's a whole kingdom of stuff. I can point to a protist. What's its definition? What comes to mind for you? You've all learned. But what's a favorite protist? Give me an example you probably learned about. I know you're smiling. What's your favorite protist?
I like amoebas.
Amoeba, yeah, who doesn't like a good amoeba with those cute little pseudopodia? Okay, and paramecia, and your favorite flagella, euglena. Okay, so great, euglena, it's got chlorophyll, paramecium doesn't. Hard to define, so we go with, okay, they're mainly singular cellular, they're all eukaryotic, and then we start running out of common characteristics. And then the guys start saying, well, some of them live in colonies, maybe we should call them protoctista. So it is a crazy kingdom. And what makes it crazier is that it gives rise to the fungus, the animal, and the plants. So there's characteristics that overlap. If you want to make a big difference, and if you want to work with a great protozoologist, my friend in Brazil is one, I'm not. There's still a lot to learn about protists. Yes.
Don't people make the argument that protists aren't really kingdoms since they're
Yeah, there's that argument, and then there's all these other people saying, yeah, but look, where else do they fit in? And it's also, you notice the algae, they'll flip. Well, they're plants, they're protists. Well, now they're plant and protist. So, yeah, and even going at genetically, trying to find those ancestors and so forth is really tough. A good biologist to learn from and to talk to, or at least learn from, Susan Katz, if you like protozoology, K-A-T-Z. Doing a lot of work in there. If you're looking for a term paper or something, Susan Katz, I think in Massachusetts. I've been talking a lot with her postdoc on different ideas also, but Katz, trying to tackle that question of paraphyletic and all that. So Margulis, God love her, she recognizes the kingdom. I believe that she's in kingdom. She's okay with the domain, but she's kind of sitting here. And what we see from this, it's kind of hard to see, but we got some kind of common ancestor. There's the archaebacteria in her projection of things, the eubacteria, the protists, the protoctista, as she calls them, all over here, and giving rise then to the fungus. The animals, look at all the names. Oh man, we know a lot about animals, and you see names up there you recognize, and then the plants. You're saying to yourself, do I have to know all this? You should know this kind of thing, because we know that Margulis is known for what big theory that we've already talked about. Endosymbiosis, very good. Eric, you are on a roll today, good for you. But so she's not only saying, and you can look through here and find over here the chloroplast. Okay, she's also saying, well, you know, maybe some of the, then also the proteobacteria making the eukaryotic cell with the mitochondria, okay, she's also saying, you know what, I think the spirochetes may have given rise, you know, gotten together into that crazy 9+2 cross-sectional linkage that makes up the fibers of the eukaryotic flagella. Well, they're kind of squiggly, you know, they maybe could do it, and we'll learn that they have an internal flagella and an external flagella. They could pull it off, you know, and Margulis thinks that symbiosis explains a lot of things. So, symbiosis. Domains, bacteria, archaea, eukarya. Okay, Carl Woest, UIUC. Never found Carl Woest when he was alive to be an appreciably approachable person, but I've had former students that went there and took classes with him and thought it was just the cat's tail. So, good, I'm glad. Carl Woest, University of Illinois, Champaign-Urbana, starts putting together this idea. I've talked about it several different times, and he classifies life differently. You know, he knows about the kingdom. He knows all that stuff. He starts saying, like, let's look at a different molecular clock. Let's put this together a different way, and he is finally given a lot of credit over time. And basically, they come up with this roadmap to life. We call it the tree of life. Now, this is the simplest tree. Those of you on the web, either now or at some point, Google tree of life. I mean, you can find like stuff you didn't even know existed, way beyond tardigrades and everything else. But we see that there's a relationship, and this relationship comes down to, in this tree of life, based on molecular work. Now, there's just, you know, this kind of mysterious common ancestor out there. Whether we will know that or not, we don't know. Alex sent me an article that, you know, there might be viral fossils from Mars. I mean, how do you figure out a virus? Wasn't that in your article?
I think something.
Yeah, it's like, how do you, you know, every once in a while we get a meteorite and go, oh, those look like bacteria. How can you tell a bacteria fossil? I don't know. Guys are much smarter than I am. But, you know, were we inoculated? Did life evolve here? We can talk about it, who knows. But it still begs the question, they had to evolve someplace. And there's a lineage. And there's mutation. And, you know, things are changing. And molecules record that. And that recording is handed on through the generation. That's how this works in a nutshell. Am I going the wrong way? Oh yeah, so what are we left with? You know, a very simple kingdom of class, order, and species, okay? Bacteria, 12 to 15 phyla that are pretty nailed down. Now there's about 39 described. And as I think I talked to you about in the first day, but we were all new together, you know, possibly even 2 more subdomains that are being found amongst the nanobacteria, you know, kind of stuff that's really small in the deep ocean. So the story is definitely open. The archaea, 3 phyla, well, that's and eukarya, well, we recognize those groups, but most everybody here had to get off the bus and get on this train, okay, and classified a little bit differently. And a lot of people kind of take it back. Wait a minute, you know, I'm a person in here, and I'm more related to a slime mold. You're putting me with that, and you know, I'm with all these other guys. I'm not my own kingdom, and this is an animal. Now get used to it. You know, prokaryotes dominate if we start looking at species. Hang in there, we've got 7 minutes. This will be fun. So let's talk about the 16S. Tree of life based on the small subunit RNA. We've all talked about, let's get a little bit in our notes, so that come exam day, we don't have long faces, we're all happy. Yay. So, small subunit rRNA. We've talked about Svedberg units. If that's foggy, look that up. That's what the S means. Okay. Prokaryote ribosome. It's got a large subunit, 50S, and a small subunit, 30S. Add it up? No, not 80, 70. Why? Go back to the Svedbergs and the centrifugation density versus volume argument on those. Okay, 18S with eukaryotes, it still works. It's from the small, ribosome, the small unit, the big unit, protein centers. All right. For those of you who like to do molecular stuff, the ribosomal database out of Eastern Michigan, yeah, is the place to look at stuff for bacteria. Although GenBank is really good. These guys are really specialized. So if you need to see how it looks like, we have ribosomes, the big subunit, two RNAs with 32 different proteins. Small subunit, 1 RNA, 21 proteins. Okay, I don't care so much about these numbers, just know it's WIC protein. You know, and if we look at the small subunit, has the 16S. Okay, about 1,500, 1.5 kilobase. Okay, and doing that molecular cloning, you can get really close to putting in a whole insert that's about that size. The large subunit, a 23S and a 5S. Remember I told you about the 3 bears here? We started looking, and people still look at the 23S. It's not like you can't. But it was almost too much information. And the 5S, has much less information and what wasn't specific enough, especially when we were doing hybridization and stuff. Yeah. Okay, so shape. We talk about the importance of fish, what's in structure and function. Okay, you really can't change around an enzyme too much. You can't, you know, and you can make whole phylogenetic trees off of enzymes like the NAS Z enzyme or a nitrogen oxidizing enzyme. And so change occurs in other things. It just is that this isn't, the shape of this isn't really enzymatic. The shape of this is to really, accommodate tRNA and do a few things that way. So a little bit more play than in some of these more specified functions in other molecules. And this is the deal. Here, several things in this slide. One, RNA can be double-stranded, and there's single-strand areas. So we have loops, double strands, and single strands. Okay, this is a three-dimensional view of this. Okay, so that's important, and it turns out that you can map it. The other thing I wanted to tell you about is that there's different regions, and you can see 1, 2, 3, 4. Different parts of this region are more variable than the others. That's what I was telling you about, that initially we were sequencing all of this to figure out what's the same and different. And you go, well, how can you get by with Illumina? You only have a few hundred bases. Because some of these are more diagnostic areas, more variable. So you can get more information per unit nucleotides. And that's the idea there. I don't need to memorize more stuff on that. Yes, the point here just is that two-dimensional, we've all seen it, I think it's in your book. You can fold this thing up and it looks much different. So let's end on this slide. I think I can, this is a great place to end and maybe to start next time. Why use the small subunit rRNA gene as your molecular clock? That's what we're talking about here. You can substitute 16S, which is on the slide, or 18S. Universally distributed. Okay, are they in viruses? No. Are viruses really alive? Well, you've argued it. I say no, but even if maybe, we got to put them on the side. Anything that can reproduce, send along its genetic information, and is going to have metabolism and make energy, has got to make protein. If you make protein, you got to have a ribosome. So we're going to compare everything from the beginning of time, if we can, beginning of time on Earth, when life began, to now. We need a clock that's in all the organisms. So there you have it. Number one, table 5. It must be functionally homologous. Okay, you can't have a molecule doing one thing in one organism and something else in another. Keratin, not work so well, right? Fingernail, hair, kind of the same, not really doing it, and so anyway, gotta have it doing the same thing. I think you get it, and move on. Okay, you have to have the ability to align it. If you have information, if you're trying to compare two articles, and you can't align the information, you can't line up the facts, it's gobbledygook, it's confusion. You can't come to a conclusion. With this, we can line them up. Maybe Dr. Bonner can talk to you about clustol, but we can take different species, part of the 16S, and say, oh, look, there's something different here, okay, and so forth here. This doesn't have, sometimes there's big gaps, but you can line it up. How can you line it up? Because there's areas that are conserved that hardly ever change. I'm going to make it up in this case. This looks pretty conserved through here, okay? And I don't see a lot of changes. Probably there's another region in here. Okay, so you can use those as your aligner, and then say, oh, in between those conserved regions, those are the variable regions. That's where I can see where things really were different. And then you can work on mathematical models on similarity. Yes, it's not sex. It comes down to nucleotide similarity, and that's a criticism, but also a strength. 4, hang in there, we're almost there. You got to have something that's got change that can record the length of time that you want to measure. We're talking about 3.8 billion years. Things that evolved from the first sulfur organism using iron sulfide as its proton motive force. We'll talk about that, but it's got to be in step with that. You want to compare a bunch of monkeys? Species, maybe cytochrome is good, or hemoglobin. People do that. Oh, 4 of them. Okay, 4 things. I thought there was a 5th, but we get that one free. See, some of you, anytime today, anyone planning to come to lab past 7? Okay, anyone planning to come after 8? No, 8 o'clock tonight. Okay, I'm going to plan to be there then from 10 to 8 p.m. So people coming at 7, I'm probably not going to kick you up. If you're doing something, we'll be there. But, you know, missed supper last night, I'm going to get supper tonight, that's all. So, coming down, survival of not the fittest, of me. See you later. Look up.